Antibodies are proteins which are made by specialized immune cells in our body called B lymphocytes. Antibodies bind and neutralize antigens which are parts of the pathogens invading our body. Once our body is invaded by a pathogen, such as a bacteria or virus, specialized cells known as macrophages engulf these and chop them down into tiny pieces. These tiny pieces are expressed to other cells called T lymphocytes. Once the T lymphocytes see these, they become active and activate B cells to produce antibodies. Once the antibodies are produced, they go around and bind to their targets, facilitating their removal by phagocytic cells. Now, where do monoclonal antibodies fit in? If we have factories in our body to produce antibodies, can we do the same in our laboratories and then use them to fight disease and vaccinate people? For some time, nobody was sure how to manufacture antibodies at a large scale. This required very large numbers of a specific B cell, all secreting identical antibodies. One main obstacle faced scientists is that B cells, which can divide by mitosis, do not produce antibodies, while plasma cells, which are antibody factories, cannot divide. The breakthrough came in the 1970s using the technique of cell fusion. A small number of plasma cells producing a particular antibody were fused with melanoma cancer cells that divide uncontrollably. The cell produced by the fusion of a plasma cell and a cancer cell is called a hybridoma. This cell can divide by mitosis and can also secrete antibodies at a large scale. Monoclonal stands for single clone, so monoclonal antibodies are specific antibodies produced by a single clone of hybridomas and consisting of identical antibody molecules. To get plasma cells, firstly, a specific antigen is injected into a mouse. The mouse immune system will produce antibodies specific to this antigen. These antigens are then made by specific B cells which are present in the spleen. These are then isolated and cultured in the lab to acquire the wanted B cell. It is then fused with a cancer cell to form a hybridoma, which can then divide and give us large numbers of monoclonal antibodies. They have many uses in diagnosis and treatment. The most important of these is their application in cancer research. They can be used to locate cancer cells which have antigens in their cell membranes that differ than the antigens on normal body cells and can therefore be detected by antibodies. Also, an anti-cancer drug can be attached to the monoclonal antibodies, which can then accurately locate and target cancer cells, thus avoiding human cells. Furthermore, they can also be used for detection, where they carry special markers which can then show doctors where cancerous cells are present. To add on to that, they can be used to trigger the immune system to recognize and then destroy the cancer cells. Monoclonal antibodies can also specifically identify other diseases, speeding up the choice of the most appropriate treatment for the patient, and can be used to locate blood clots in the body. As the biological sciences are developing, so is the range of monoclonal applications, as they are now being used in the treatment of our recent pandemic, and who knows where they will be used next.